Jai Shri Krishna everyone. My name is Rakshitha Pramod Nesur. Currently I am pursuing master degree in chemistry at JSS College of Arts, Commerce and Science, Ooty Road, Mysuru. In today's video I am going to explain about what are the principles of colorimetry in analytical chemistry, structure and parts of colorimeter and what are the applications. So let's know what is colorimetry by taking an example. Here I am taking an test tube which is consisting of copper sulfate solution. As you know, copper sulfate solution is in blue in color, so that's why I am taking here the blue color solution. These are the incident light. These are parallel and monochromatic. Monochromatic is means the single color. As I pass these monochromatic parallel rays through the solution, few of them get absorbed by the solution and other will get transmitted. The light or the rays which are get absorbed by this solution, that is known as the absorbance or also known as the optical density. You know that white light is belongs to visible spectra, which is having 400 to 800 nanometer of wavelength. White light is not a monochromatic because it's consisting of all the light which are present in visible spectra like a violet color, blue, green, yellow, and orange color. If I pass a single color, then it will become monochromatic. Then what is the transmittance and what is the percent transmission? The transmittance is nothing but the ratio of the transmitted light and the incident light. Transmitted light to incident light, and percent transmission, which is equal to intensity of transmitted light by intensity of incident light into 100. Always remember that these transmitted light are having lesser intensity than the incident light. Let's understand what is the Beer's law. Again, I am taking here three test tube, which is first one consisting of 100 mg percent of copper sulfate solution, second one of 200 mg percent of copper sulfate solution, and third one is of 300 mg percent of copper sulfate solution. I am taking here incident light, which are parallel and monochromatic light. When the light passes through the solution, the few of the rays get absorbed by this copper sulfate solution in three test tubes also, and next other get transmitted. As you can observe here, the number of the amount, or you can considering as a number, the number of transmitted rays get decreases. In first test tube it's more in number, in second test tube it decreases, in third it also decreases. From this we conclude that as we increases the concentration, as we increases the concentration, the absorbance will also increases. This is the Beer's law. When parallel rays of monochromatic light passes through colored solution, its optical density is directly proportional to concentration of colored solution. Okay. Optical density is nothing but the absorbance. As we increases here concentration, the transmitted light rays get decreases and the absorbent get increases. This is the Beer's law. Optical density is directly proportional to the concentration. Okay. Now let's understand what is Lambert's law. Here again I am considering three test tubes of different diameter. First one is of 1 centimeter, second one is of 2 centimeter, and third one is of 3 centimeter. As I already said, I am taking here the concentration of copper sulfate solution. In these three test tubes, we are pouring same amount, 100 mg percent of copper sulfate solution. But there is a difference in only their diameter. Okay. As I passes the monochromatic parallel light through this solution, few of the rays get absorbed and few of them get transmitted. As you can observe that here, the transmitted rays number get decreases after increasing the diameter. Then the Lambert's law explained when a parallel rays of monochromatic light passes through colored solution, its optical density is directly proportional to path length of the test tube. So optical density is directly proportional to the path length. What we have studied before, we are just applying in the instrumentation. I am taking here the light source. As a light source, the tungsten bulb I am taking here. Why the tungsten bulb? Because it emits all the lights which are present in the visible spectra. As I already mentioned before, white color, blue, green, yellow and orange, which comes under the range of 400 to 800 nanometers. That's why I am considering tungsten bulb. Next, I am placing at the proper distance a convex lens because the lights which are get emitted by these tungsten bulbs, these are not a parallel. To convert them as a parallel rays, I am placing at the proper distance convex lens. After that, we are placing here the monochromator. In this place of prism, I can also place the colored glass. The colored glass. If I wanted the yellow color, then I will place the yellow colored glass here. That is the filter. This is also a filter. We can place both prism or colored glass. After that, we are placing prism just to convert these light which are parallel into monochromatic light. Okay. After that, we are placing the cuvette. Why the cuvette is placed here? Why not the normal test tube is placed? Why? Because the normal test tube is having the air bubbles, micro air bubbles. They will be distort the light radiation directions. So, right, that's why we are using cuvettes. Cuvettes are also scratch durable and these are parallel. As you can see, these are parallel. So, that's why cuvettes are used in place of the normal test tube. After that, we are placing the photocell here. Photocell, it is nothing but the silicon or the solar panel which convert the solar energy into electrical energy like that. We are placing the solar photocell which is directly connected to the voltmeter or the voltage. Which convert the light radiations into voltage. After taking these readings by applying any mathematical formula, we can calculate the concentration of the light which we are desired. These are the points as I mentioned, as I just explained. And let's know what are the applications of this colorimeter. What are the applications now? The concentration determination, chemical analysis, quality control, and environmental monitoring, biomedical research. In all these sectors, we can use the colorimeter. But the main application of this colorimeter is to concentration determination, which is used to measure the concentration, mainly the concentration of substance present in the solution. And also in the chemical analysis to analyze the presence of quality of specific compounds and to monitor the product quality and consistency in industry like foods, pharmaceuticals, textiles in the food like beverages, and environmental monitoring to measure water and air quality by detecting the pollutants concentration. And next is to study biomolecules such as the proteins and nucleic acids. So now we have understood what are the applications, how the colorimeter works, and what are the principle behind it. This is all about the colorimeter and its principles in analytical chemistry. Hope you all get that. Thank you.